Okay, then this is a quick session on epistasis, another a type of inheritance or pattern of inheritance that involves two or more genes contributing to a phenotype. Okay, so we'll say what it is. I'll give you a bit more of a definition there, and apply the concept of epistasis to some unfamiliar contexts. So this is on page 18 in the notes there. Epistasis. Okay, so as I said before, when two or more genes contribute to a particular phenotype, we call this epistasis. So the idea is that one allele of a particular gene can affect or mask the expression of another one. This often happens in a metabolic pathway. So metabolic pathway is a series of reactions that take place one after the other. And in that metabolic pathway, those reactions are controlled by enzymes. And the enzymes obviously require a substrate. So one reaction takes place. Let's have a look at this diagram down the bottom. Let's imagine the first reaction is this reaction from the substrate. Each of these are substrates. Substrate A being converted into the product B by enzyme number 1. Product B then becomes a substrate for enzyme number 2 and is converted into product C. C then becomes a substrate for enzyme number 3 and is converted to the final product in this pathway, D. Okay, so if an allele for one of these enzymes produces an enzyme which is non-functioning or different in some way, the substrate, so if the allele is different, the tertiary structure of the enzyme will be different, the active site will no longer be complementary to the substrate, and therefore one of these steps may be affected. So if we imagine that there are two alleles for enzyme number 2, the dominant allele and a recessive allele. Now the dominant allele codes for enzyme number 2. So an individual who possesses one of those dominant alleles, so is either homozygous dominant or heterozygous, is able to produce enzyme number 2 and is able to convert substrate B into product C. However, if an individual were homozygous recessive for this A allele, this enzyme would no longer function. So substrate B could not be converted into substrate C, and therefore the final product D cannot be produced either. Let's have another look at an example of this. Okay, so in this example, this is the one that you've got in your notes. There are two genes, gene A and gene B. Now, gene A has two forms. It has the dominant allele and the recessive allele. Gene A, or the dominant allele of gene A, codes for enzyme Y. Anyone who possesses gene A, so anyone who is homozygous dominant or heterozygous for gene A, is able to produce enzyme Y. They're able to convert substrate, this white substrate, into a pale blue pigment. That pale blue pigment can be converted into a purple pigment if the active enzyme Z is present. There are two alleles for this gene. Dominant allele B produces an active enzyme Z. The recessive allele B produces an inactive one. So a person who was, let's add these two things together now, either homozygous dominant, or heterozygous for A. So I'm just going to put a little space in there to say that they could be either of those two things, and possesses either, at least one dominant allele for B. Well, they can produce enzyme Y, converting the white substrate into a pale blue pigment. They can also produce the enzyme Z, which takes the pale blue pigment into the purple pigment. So if we think about these phenotypes here, the phenotype is what the final colour of the pigment will be. So an individual who is homozygous dominant for both. Homozygous dominant means they can produce allele, they possess the dominant allele A, so they can produce Y, white into pale blue. They've also got a dominant for B, so they've got dominant allele B, active enzyme Z, pale blue into purple. So that individual would have the purple pigment. Likewise, for individual B here, they've got a dominant A and a dominant B, able to produce both of those enzymes. So also purple. This person here has a dominant A. They can produce the active enzyme Y, taking that white substrate into a pale blue pigment. However, they've got two recessive alleles for gene B. So they've got inactive form of enzyme Z, so they will do the first step but not the second one, so they remain a pale blue pigment. This person here, well, they are homozygous recessive for gene A, so they've got the inactive form of gene A, so there's no enzyme Y can be produced. They would remain white. And then this last one, this might, you probably worked it out already, but this is sometimes where students trip up. Okay, they have homozygous recessive A alleles, so they've produced an inactive form of enzyme Y, or they don't produce enzyme Y at all. So the white substrate is not converted into pale blue pigment. So it is, it doesn't matter that they could potentially produce this second enzyme with the dominant allele B, because there will be no substrate for that second enzyme to act on. So these people remain. Excuse me. These people would remain. I say people. The white substrate would remain. Okay, let's have another look at a different example. Okay, so these are mice. In mice, gene A controls the distribution of melanin, which is another type of pigment. That melanin on the mouse hairs, the dominant allele A leads to black bands forming. The recessive A leads to uniform black hairs. Okay, so the hairs themselves can either have black bands that look like this, or they can just be solid black. That should be solid black. I've not coloured that in very well. Okay, so if they possess a dominant A, they will have these black bands referred to as agouti. If they are homozygous recessive, uniform black hairs. So this is about the distribution of the melanin. Gene B controls the colour, which determines the expression of gene A. So a dominant B leads to the formation of melanin. Let's think about that, that's important. Whereas a recessive B leads to no pigment at all. So essentially what we're saying here is it's necessary to have a dominant B to have any melanin at all, and then the genotype of this A gene determines whether there are bands or no bands. So if an agouti mouse, homozygous dominant for both genes, is crossed with an albino mouse, homozygous recessive for both genes, what will the genotypes and the phenotypes be? So what it's saying here, homozygous dominant crossed with homozygous recessive. Okay, only gametes that can be produced here are both dominant. The only ones here are both recessive. All of the offspring will be heterozygous for both. So 100%, something like that, and they will all be referred to as agouti. Okay, they've got the dominant A for the bands for the agouti, and they've got the dominant B to produce the melanin in the first place. The second part of this question, if two of those offspring from question 1, from this question, so the two of these offspring were crossed, what will the genotypes and the phenotypes be for the F2 generation? Okay, so we've got heterozygous genotypes crossed. Now, because they're heterozygous for two genes, there are 4 possible gametes. So big A, big B, big A, little b. That's a little a, big B, little a, little b. In those circles to show their gametes, and it will be the same for this. So what we're going to end up with is a dihybrid cross that looks something like this. Here are the 2 heterozygous parents. Here are the gametes crossed with each other, and then our Punnett square to show the outcomes. Okay, what we find is there are 9 agouti, 3 black, and 4 albino. So it doesn't quite follow that 9, 3, 3, 1, because if there are 2 recessive A's, there will be no bands either way. Okay. Last little bit we'll do on this is sometimes epistasis is an important factor for regulating the amount of product produced. So in some cases, one of the products, perhaps the end product of the metabolic pathway, acts as an inhibitor to the enzyme or an enzyme earlier in this sequence. The importance of that is it prevents the cells from producing too much of the end product, because that end product goes back, it inhibits one of these first enzymes, and that regulates the production of the intermediate compounds and therefore regulates the production of that end product. Then, if they've been inhibited for a while, the concentration of the end product would decrease, there'd be less inhibition, and the process can continue. Okay, in your question booklet, you've got questions 20 and 26. Could you have a go at completing those for me? Thank you.